Okay, so so basically, signal transducts and pathways look like a really complicated wiring diagram, and they have, and probably 5 or 10% of all the components of a set of the genes are involved in encoding components that control these signal transducts and pathways because they play such an important role. And since there's so many of them, the key strategy is where to start in this, when you want to work on signal transducts and pathways. So I've always started to work on things that are strongly linked to human disease, and those are the things that my laboratory works on, and I'll talk about 3 examples of this in my talk. But before I start telling you about the work I do, I just wanted to tell everybody that about the principal components of this pathway, and one of them is protein phosphorylation, and the other is protein ubiquitilation. And enzymes that regulate protein phosphorylation and ubiquitilation lie at the heart of these signal transducts and pathways. So I'm sure you all know that protein phosphorylation is mediated by a protein kinase phosphorylating a substrate, and the phosphorylation of the substrate can influence its function in virtually every conceivable way. It can activate it, it can inhibit it, it can change its localization, influence the way it interacts with binding partners, and probably protein phosphorylation forms the key backbone of the signal transducts and pathways. And this list is not exclusive. This is from the genetic analysis of human disease. It's become very clear now that mutations of protein kinases, inherited mutations in diseases in protein kinases really control many familially inherited disease. And I've highlighted a few on this slide that kinases that my lab particularly works on. But phosphorylation now has a rival in protein ubiquitilation. So there's about 530 protein kinases, and it turns out that there's at least as many enzymes that regulate ubiquitilation as they are that regulate protein phosphorylation. And protein ubiquitilation is a similar process. It involves an enzyme called an E3 ligase putting ubiquitin chains, either single ubiquitin molecules or ubiquitin chains on substrates, on the lysine residue and substrates, and this can also influence the substrates in many different ways. For example, one particular type of ubiquitin chains that put on substrates called lysine 48 link chains mediates degradation of the substrate, but similarly ubiquitination of substrates can result as recruiting platforms for other signaling molecules to bind to them, or it can influence their ability to interact with other signaling proteins. And I just wanted to say as well that it's also become clear that there's as many components of the ubiquitilation system that are mutated in human diseases as there are in protein kinases. Here I've just listed some of the enzymes that are mutated in E3 ligases. These are the enzymes that put on ubiquitin chains. Also the enzymes that take off ubiquitin chains, the deubiquitilases, are also significantly mutated in human disease. And there's a lot of proteins that have ubiquitin binding domains that recognize ubiquitilated proteins, and these are also heavily mutated in different human diseases. So and then there's a lot of interest now at exploiting our understanding in protein phosphorylation and ubiquitilation in the drug discovery process. And I thought it'd be quite interesting just to compare the 2 modifications side by side. Obviously the protein of phosphorylation area is much more advanced than our understanding of protein ubiquitilation. But the key points are that we have as many enzymes that regulate protein phosphorylation as ubiquitilation. The phosphorylation and ubiquitilation is a reversible process. That this enables it to be a signaling system. It can be switched on and switched off. And we also have a similar number of enzymes that reverse this ubiquitilation. And and the first drug in the phosphorylation area was Gleevec, a drug to treat CML leukemia, and this has revolutionized actually the treatment of CML patients, and now patients are actually nearly cured by staying on the Gleevec drug, and this has resulted in an explosion in the field of protein phosphorylation, and currently there's 25 drugs that have been approved that inhibit diverse protein kinases. There's over 150 drugs in clinical trials, but by contrast, in protein ubiquitilation, there's only one drug that that's been approved, and This is bought as Zenabib or Velcade. This is a proteasome inhibitor, but that one drug actually still makes $2.7 billion sales, mainly for blood-driven cancers, and there's many fewer compounds in the clinic in the area of ubiquitilation. And given the importance of protein phosphorylation, we actually have 30% of all research and development in pharmaceutical companies is currently aimed at trying to develop compounds that inhibit protein kinases to try and restore normality to signal transduction pathways that are disrupted in causing human diseases. But the expectation is that as we do more research in the area of ubiquitilation, we'll understand more, and this will result in new ideas to treat disease by targeting the ubiquitilation system. So in my talk today, I'm going to give you 3 examples of signaling pathways that my lab works on. The first one is going to be the Wnt signaling pathway that regulates blood pressure. The second one I'm going to tell you about a signaling pathway involved in Parkinson's disease. And the third one I'm going to tell you about a signaling pathway involved in cancer. And I suppose the themes of my talk, at least the first 2 sections of my talk, will be understanding how ubiquitilation pathways regulate kinase signaling pathways, and we'll see some interplay between phosphorylation and ubiquitilation that I think is going to be a major theme in understanding signal transduction pathways. And I also hope to try and emphasize some ideas that can come out from our research that gives us new ideas and how to treat these diseases in the future. So the Wnt signaling pathway. So this area of research, like many areas of research that I work on, starts with fantastic genetic analysis of human patients. So Ritzer Lifton discovered that there's a hypertension syndrome called Gordon syndrome, where patients have severe hypertension, severe hyperkalemia, that's high serum potassium levels, and he discovered that patients with this Gordon syndrome had mutations in 2 unstudied protein kinases that are called Wnt1 and Wnt4. So these are gigantic kinases. I won't go into too many details, but they have a kinase domain at their N-terminus and about 2,000 amino acids, threads of amino acids at their C-terminal tail. And at least for Wnt1, it was quite clear how the mutations caused the disease. There are always intronic deletions within the Wnt1 gene that slightly elevated expression of the Wnt1 messenger RNA. So the idea was that patients with these mutations had too much Wnt1 being expressed in their body, and this somehow led to blood pressure. When I first read these papers, we always thought in signal transduction as being involved in cancer, diabetes, inflammation, neurodegeneration, but I never could understand how a protein kinase could regulate blood pressure. So this is sort of the primary motivation for getting into this field in the first place. And the next slides are going to sort of summarize about 10 years' work that my laboratory has done to understand how the Wnt signaling pathway operates in cells. And then I'm going to tell you about some new advances in this signaling pathway. So basically, what we've discovered is that the Wnt1 and Wnt4 kinases, they're actually activated by, they sense hypotonic low chloride conditions in cells and osmotic stress. And they're very potently activated, especially when you reduce the chloride levels in the extracellular medium. This results in somehow, by an unknown mechanism, a dramatic activation of the Wnt protein kinases. And then what they do is they phosphorylate and activate 2 downstream protein kinases that are called SPAC and OSR1, and these kinases become activated after the Wnt's phosphorylate them. And then, very interestingly and relevant to hypertension, what we discovered in the kidneys, this is in the distal convoluted tubule or the thicker sending limb of the kidney, the SPAC and OSR1 phosphorylate these very critical iron co-transporters that play very important roles in transporting ions from the urine back into the blood. And these are either the NCC, co-transporter that's actually the major drug target for the frontline treatment for hypertension, which is thiazide diuretics, or NKCC2, which is the drug target for the loop diuretics. And basically what this pathway does when it's active, it stimulates the activity of these iron co-transporters. So when Wnt is overexpressed in these patients, what happens is the SPAC OSR1 kinases are too active, these sodium co-transporters are too active, these patients therefore retain too much salt by transporting too much salt from the urine into their body, and retaining too much salt results in the hypertension. And consistent with that, actually, the patients of Gordon syndrome can be treated very effectively with thiazide diuretics. They're actually sensitive to this drug, and you can use one-fifth of the normal dose of the drug to reduce blood pressure in these patients. And we've done a lot of work to confirm this pathway is correct. For example, we've made mice in which we can't activate SPAC and DOSR1, and these knock-in mice that this pathway can't be switched on have very low phosphorylation of these co-transporters. They also excrete too much salt, and they have, low blood pressure. Interestingly, the key phosphorylation sites on NCC that are phosphorylated by SPAC and DOSR1 are frequently mutated in patients with Gittelman syndrome, which is a condition which results in low blood pressure. Because if you mutate these phosphorylation sites, SPAC and DOSR1 can't activate these co-transporters, and this results in too much salt being excreted, and this results in low blood pressure. So this pathway was sort of looking quite well understood, and we were actually starting to wonder whether we should find a new project to work on when about a year and a half ago these two papers appeared that suggested that there were a significant number, I think about 50 or 60 other Gordon syndrome patients had been identified in which the mutations weren't in the Wink1 or Wink4 genes, and instead they found that mutations were present in regulatory subunits and the catalytic subunits of an E3 ubiquitin ligase. So if I was amazed that protein phosphorylation could regulate blood pressure, I was even more astonished to find that ubiquitilation, mutations in a component that regulates ubiquitilation could also be involved in regulating hypertension. So, this work I'm going to tell you about was done by, in collaboration with, was done by Kito in my lab, Axel Niebel in our unit, and also in collaboration with Timo Kurtz's laboratory and his postdoc, Francis Rose. And, this concerns, understanding how these components, this is a Cullen E3 ligase KLH complex, were involved in regulating Gordon's hypertension syndrome. So, the genetic data suggested that mutations in these relatively unstudied components caused hypertension, but it wasn't known how these things, operated or functioned. So it turns out that all E3 ligases, and most of them, like this is the catalytic subunit, it's called Cullen E3 ligase, is a catalytic subunit, but they need a regulatory adapter subunit to operate. And this regulatory adapter subunit in this case is called KLHL3, and its role is to interact with substrates. It has a substrate binding component, and this is critical for enabling the E3 ligase component to then ubiquitilate the substrate. And often with Cullen ring E3 ligases, this ubiquitilation occurs in lysine 48, and it triggers the substrate to become degraded by the proteasome system. So the key thing was to understand how this was connected to blood pressure was to try and identify what the KLHL3 subunit interacted with in the cell. So we did a fishing experiment where we took cell extracts, and we tried to absorb proteins from these extracts that could bind to wild type KLHL3, and we also used a form of KLHL3 that was mutated in the patients with Gordon's syndrome. And what we found actually was that we could absorb to these KLHL3 subunits, one major high molecular weight band was absorbed from the lysates when we did this fishing experiment, and this turned out very satisfactorily to comprise all the major Wink isoforms that are present in the cell. So I'll just go into a bit more details now. So this is the molecular architecture of the KLHL3 subunit. It has a BTB in the back domain, and this region of the protein regulates binding to Cullen 3. It also has 6 Celts-like repeat domains, and this is known to regulate binding to substrates. And you can see that the human mutations that cause Gordon syndrome either lie in the in the domain that binds Cullen 3 or the domain that's predicted to bind to substrates. So the prediction would be that the mutations in this region would stop binding to Cullen 3, and the mutations in this region would stop binding to Wink1. And the experiment that we did that shows that this is the case is shown here. These mutations stop binding to Cullen 3, whereas the mutations in this region most of them, inhibit or strongly, or ablate interaction with the Wnt isoforms, but not to colon 3. So this experiment suggested that what was going on, was that the key substrate for this E3 ligase complex was the Wnt1 protein itself, and the binding of KLH3 to Wnt1 would target it for ubiquitilation, and therefore mutations in these components that are found in human disease would inhibit this ubiquitilation or inhibit assembly of this complex, and this would result in too much Wnt1 being expressed in the cell, and this would be the cause of the hypertension in these patients. So we then set up an experiment to see whether this complex could indeed ubiquitilate Wnt1. This is an in vitro experiment. You actually need 6 components in this reaction to ubiquitilate Wnt1. You need these 4 components here. You also need an E1 ligase, and you also need ATP. And so we had to make all these 6 components, and we needed to add, immunoprecipitated Wnt1 into the reaction mixer. So it's actually quite a heroic experiment to do this. But it turns out when you add all 6 components to this reaction mixture and you add, ATP, you can see a very dramatic ubiquitilation of Wnt1 in the assay. And if you leave out any one of the 6 components, you lose this ubiquitilation. And also very satisfyingly, if you do this reaction with the disease mutant form of KLH3, this is no longer able to induce the ubiquitilation of Wnt1 in the reaction. So that was quite satisfying, but there was one thing we didn't understand in the Wnt signaling pathway. So we understood how mutations in Wnt1 caused hypertension, because these were intronic deletions that increased expression of the Wnt1 messenger RNA. But it turned out that the mutations, many patients with Gordon syndrome also have mutations in Wnt4 protein kinase. And it turned out that all the patients with mutations in Wnt4, the mutations lie within a non-catalytic region of the protein, in no man's land, in this region of the protein. And all the mutations lie within a 3 amino acid or 4 amino acid sequence in a very basic acidic region of the protein that's highly conserved between Wnt isoforms. And we could never understand how these disease-causing mutations influenced Wnt4, because all the biochemical experiments that we did suggested that these mutations did not influence the catalytic activity of Wnt4. But everything became crystal clear when we asked the question of where does KLH3 bind to, because in Wnt isoforms, because we know obviously KLH3 recognizes Wnt and binds to them, but we didn't know what the binding site was. What's the degron motif that's critical for mediating the interaction between Wnt isoforms and KLH3? And when we did this, when we did the detailed experiments to identify the key binding site for KLH3, what was astonishing was that it turned out that the KLH3 actually binds to this same 9 amino acid sequence that is mutated in Wnt4. And then it became crystal clear why these mutations caused hypertension, because we predicted that these mutations would work not by influencing Wnt4 activity, but instead by disrupting the ability of Wnt4 to bind to the E3 ligase that disrupts its, that catalyzes its ubiquitilation. And so we did some experiments to verify this. I don't need to go into all of the details here, but very satisfyingly, when you introduce these disease-causing mutations, for example, this is the glutamic acid 562, or this is a K565 residue. When we introduce these disease-causing mutations into Wnt4, this stops their ability to bind to KLH3. So this was and more recently, in collaboration with a group of Alex Bullock and Fiona Shorell at the University of Oxford, we've been able to crystallize the KLH3 bound to the Wnt4 degron motif. And it turns out that the 6 KLH cell repeats form a sort of a bowl-like structure with fingers that point up, that form very exquisite, specifically interactions with this 9 amino acid, acidic region. And what's very satisfying is that many of the key interactions that enable this degron motif to bind to KLH3, for example, the this arginine 528 residue, they bind directly to this aspartic acid residue that's also mutated in disease. So in humans with mutations in this residue or this residue, you get hypertension. Also mutations in this residue, and either one of these two residues also cause hypertension in humans. So I think this is interesting because it suggests that basically, all this hypertension is caused by mutations that either increase messenger RNA levels of Wnt1 or missense mutations in Wnt4 that prevent them interacting with the E3 ligase, or mutations in the E3 ligase components themselves that mediate this, this ubiquitilation. And I think in this pathway, what happens is that in the kidney every day we transport many kilograms of salt from the urine back into the body, and even if the Wnt signaling pathway is, let's say, 5 or 10% overactive because of overexpression of the Wnt signaling components, this will result in the pathway being too active in the kidney and will result in too much salt being reabsorbed, and this will cause complications because the other backup systems in the body will no longer be able to compensate. And this is the principal cause of hypertension. I think it also illustrates the point that when we start doing a lot of analysis of human disease and sequencing many patients, we'll probably find more and more examples of mutations that occur within non-obvious regions of your protein that don't affect the catalytic function, but they lie in what looks like unstructured non-functional motifs around the protein. And I think many of those will actually lie within the degron binding motifs that regulate the stability of a protein. So I think it'll be very important to, when you're looking at and trying to understand, how mutations affect protein function, that one takes into consideration that the mutations may actually lie in sequences that bind E3 ligases, and therefore mutations in these critical residues will have very profound effects because they will result in your protein being too expressed and therefore becoming too active, and causing human disease. So the second thing I'm going to tell you about is the Parkin signaling pathway, and this work is mainly done by my colleague Miratul Makit, and we have a joint PhD student whose work I'm going to talk about, Agne, who's mainly based in Miratul's lab, but I also participate in her supervision, and this is the sort of very interesting story that I'm going to tell you now about a very unexpected role by which this PINK1 kinase is involved in Parkinson's disease can activate an E3 ligase called Parkin that's also involved in Parkinson's disease. So the reason we're really interested in Parkinson's disease is because beautiful genetic analysis that's been done, much of it's actually been done in the UK, characterizing patients with Parkinson's disease, has led to the identification of about 20 familial genes whose mutations in humans causes either early onset or normal onset forms of Parkinson's disease. And many of these genes encode signal transduction components. For example, in green, I've got components that regulate the ubiquitilation system, or protein kinases, which are in red. And we really understand virtually nothing about how these components function, how they work, how they operate. And in a way, I think this list is a bit like probably when the people 20 or 30 years ago were looking at the cancer genome, mutations that cause cancer in the human genome, for example, RAS, P53, RAF. We didn't know what signaling pathways these mutations lie in. And the expectation is that many of these pathways will lie in a signaling network that plays a critical role in regulating probably survival of dopaminergic neuron cells. And it's very important for us now to try and understand more about these components and to try and understand whether or not they work in a distinctive signal transduction pathway. So so now we're going to focus on the PINK1 protein kinase and the Parkin E3 ligase. And so my colleague Miratul, he works mainly on this PINK1 kinase. It's the only known mitochondrial protein kinase. It has a kinase domain. It has a mitochondrial targeting sequence that is N-terminus, and it's localized mainly on the protein mitochondria. And then so patients with mutations in which you lose both copies of the Parkin or PINK1 gene, these patients develop early onset Parkinson's disease, typically in their early 20s. And very elegant genetic studies mainly in Drosophila, have suggested previously that PINK and Parkin may function in a common pathway, which explains why patients with either one of these mutations display very similar symptoms, in the clinic. And what previous work that Miracle did actually suggested that, the way this system was working was that, this is a pathway that recognizes mitochondrial damage. So when mitochondria are damaged, or you use chemical reagents to induce mitochondrial depolarization, you see within about 20 minutes, you see a dramatic activation of the PINK1 protein kinase. It goes from being an inactive kinase to being a very active kinase. We still don't understand the sensing mechanism behind that and how mitochondrial depolarization activates PINK1, and that's a sort of a very important question. But once PINK1 becomes activated, at the mitochondria, what it does, it then phosphorylates this Parkin E3 ligase and activates it. And the idea is that this is also located at the mitochondria, and this then ubiquitilates the outer surface of the mitochondria and targets the mitochondria to be degraded by a process of mitophagy. So this is probably an important pathway, because when that mitochondria are damaged, it probably makes sense to get rid of them from the cell, and rather than keeping damaged mitochondria in the cell. And so what the previous work, what Miracle had discovered was that the PINK1 protein kinase actually activates this Parkin E3 ligase by phosphorylating a single serine residue within a regulatory domain that's called the ubiquitin-like domain at the N-terminus of the protein. And this is the C-terminal catalytic domain. I'm not going to go into the nuts and bolts of this, but this is the catalytic domain, and this is the regulatory domain. And the critical phosphorylation site was thought to be serine 65, which is, as expected, is highly conserved throughout evolution. And, so this is what we thought, and we published a paper showing this, and, we thought the whole pathway was solved. But, then Agnew did one further experiment which suggested that there was more to discover. So before I tell you about that one experiment, I just want to show you that, how our assay works. If you take Parkin, which isn't phosphorylated, it's going to be inactive, and when you phosphorylate it, it's going to become active. And to measure activity of an E3 ligase, you can look at either auto-ubiquitilation. So many E3 ligases, when they become activated, they can phosphorylate ubiquitin chains in vitro to catalyze the formation of ubiquitin chains. You can also look at the ubiquitilation of a substrate, and the only known substrate that works well in vitro for Parkin is something called MIRA1, so you can look at the ubiquitilation of MIRA1, and you can also look at the auto-ubiquitilation of Parkin itself. So these are the three assays we tend to look at to assess Parkin activity. So this is the typical assay. If you take Parkin that's incubated with catalytically inactive PINK1, so it can't be phosphorylated, you can see you don't get auto-ubiquitilations. But if you incubate it with wild-type PINK1, you see the Parkin becomes activated, and you get this dramatic ability to induce this auto-ubiquitilation. Also, the activated Parkin can ubiquitilate MIRA1, whereas the inactive one, can't. So that's the assay we're going to use. So the key experiment that Agnew did to disprove that this was, this model was sufficient to account for the activation of Parkin was, what he decided to do is he decided to chop off this UBL domain. So if you chop off this UBL domain, surely if this model is right, the C-terminal region of Parkin must be independent of PINK1, because it can't be phosphorylated anymore. So this is the experiment that Agnew did. So see, this is the experiment I've just shown you. As he chops off this UBL domain of Parkin, he finds that the Parkin is slightly more active, probably because it's lacking this regulatory domain, and it's possible that the regulatory domain functions as an autoinhibitory domain that keeps the Parkin, less active. So when you chop off the UBL domain, and other people have shown this previously as well, the Parkin becomes slightly more active. But if the model is correct, it shouldn't be further activated when you add in PINK1. But what Agnew discovered was quite startling to us, that once he added in wild-type PINK1 to this reaction, the delta UBL domain, although it can't be phosphorylated by PINK1, still became massively activated. So the question is, and this can be also seen by the auto-ubiquitilation of the Parkin. It becomes so active that the Parkin becomes totally ubiquitilated in this region of the cell. So the question is, PINK1 must be phosphorylating something else in the reaction mixture to be mediating the activation of Parkin. So we looked in the reaction mix to see what else PINK1 was phosphorylating, and it turned out that PINK1 was phosphorylating ubiquitin itself, very, very dramatically. You can see this is actually Parkin. This is the normal phosphorylation of Parkin we see, and this is the phosphorylation of ubiquitin. And it turns out PINK1 phosphorylates ubiquitin rather well. We've tested a lot of other ubiquitin-like domains that are shown here, and these aren't phosphorylated by PINK1. Ubiquitin is phosphorylated, and the free UBL domain of Parkin is also well phosphorylated. And we've tested now 160 protein kinases that we haven't done to see which ones phosphorylate ubiquitin. And what we found is that none of the other 159 protein kinases we've tried phosphorylate ubiquitin. Well, only PINK1 is capable of phosphorylating ubiquitin efficiently in these experiments. And so it turns out that the PINK1 phosphorylates ubiquitin at a single residue, and this is called serine 65 as well. Confusingly, it's the same residue that it phosphorylates in Parkin, but it's a very conserved, serine residue that's a single site phosphorylation. And when you mutate that site, you lose the activation. And it turns out that this phosphorylated form of ubiquitin is really the key activator of Parkin in cells. So we can actually now purify stoichiometrically phosphorylated, dephosphorylated ubiquitin and fully phosphorylated ubiquitin. And then what we do is we take Parkin, there's no PINK1 in these experiments, and we titrate in either unphosphorylated ubiquitin and you get no activation, or you titrate in phosphorylated ubiquitin and you start getting activation with the phosphorylated ubiquitin. And you can also see this with the C-terminal catalytic region of Parkin. When you titrate in the phosphorylated ubiquitin, you can see you get a dramatic activation of the E3 ligase because it's binding to the phosphorylated ubiquitin. And the other key experiment we did using mass spectrometry was to demonstrate that phosphorylated ubiquitin actually exists in cells, and this is the experiment that was done. And in this experiment, we can see when you overexpress wild-type PINK1 in cells and you induce mitochondrial depolarization, you see a dramatic increase in the level of phosphorylated ubiquitin in cells, and we now have antibodies that can detect phosphorylated ubiquitin, and we have evidence that the endogenous ubiquitin is phosphorylated at serine 65 following mitochondrial depolarization. So our model is at the moment, then the way this is probably working is that once PINK1 is activated following mitochondrial depolarization, you get phosphorylated ubiquitin, and this is inducing mitophagy, and this is the technical model that we're working with, and there's more data to support this model that I'm not going to show you, but the evidence suggests that Parkin exists in cells in a closed inactive conformation, and in which serine 65 in the UBL domain is actually buried, probably by binding to the catalytic domain. So once PINK1 becomes activated in the second step of the process, you get formation of phosphorylated ubiquitin. This probably binds to the catalytic domain, maybe competing with the site that this domain binds to. And this probably then opens up the structure of Parkin and exposes the serine 65. And then we believe that PINK1 then is able to phosphorylate this serine 65 form. So whether this is the fully active form, or maybe this then folds back to this site and displaces the phosphorylated ubiquitin, that's also possible. But we think that this is the central mechanism by which Parkin is activated in cells. And I think this is really interesting because it suggests that obviously this describes a new chemical messenger by which this is able to activate Parkin. It's obviously very interesting to know more about phosphorylated ubiquitin at serine 65. Is this is this pathway really disrupted in patients with Parkinson's disease? From a therapeutic point of view, we would like to design small molecules that maybe mimic the phosphorylated ubiquitin, and maybe these would be able to induce activation of Parkin and in cells, and maybe that might have utility for the treatment of ubiquitilation. And I mentioned in my introduction that phosphorylation of there's this interplay between phosphorylation and ubiquitilation, and it's possible that that phosphorylation of ubiquitin actually is one of the starkest examples of the interplay between ubiquitilation and phosphorylation. And what's really interesting actually is if you look at the data from global mass spectrometry analysis in cells, it's quite clear that ubiquitin is phosphorylated at other residues in the cells, and we don't know anything about this type of biology and all the kinases that might phosphorylate ubiquitin at these sites, or the function that these phosphorylated forms of ubiquitin play. But I think it would be really exciting to explore whether phosphorylation of ubiquitin really plays a more general role in binding to critical signal transduction components, and does this stimulate further areas of biology. And I'm just going to be the last 5 minutes of my talk now. I'm going to tell you about a third signaling pathway that we've done a lot of work on, and this involves the mTOR signal transduction pathway, which plays a very key role in cancer signaling pathways, in which growth factors stimulate the activation of this mTOR signaling pathway. There's two waves of this pathway. Wave 1 leads to the activation of the AKT protein kinase through the PI3-kinase, PDK1 and mTOR pathways, and then wave 2 leads to the activation of this mTOR complex 1 through this TSC1 and 2 pathway. And this is also regulated by, this branch of the pathway is regulated by other conditions such as nutrient energy, mitogen stress, and oxygen levels play critical roles in regulating this second branch of the signaling pathway. And the reason why this pathway has received lots of attention is because in the majority of all human cancers possess mutations in these signal transduction components that lead to the constitutive activation of this mTOR signaling pathway. And this really plays a critical role in driving the proliferation and survival of many cancer cells. And our lab's done a lot of work to try and understand the mechanism by which this AKT protein kinase pathway is activated in cells. And and basically what we've discovered is summarized in this slide here, is that AKT is very efficiently activated by PDK1, and there's two mechanisms that drive the activation of the AKT kinase by PDK1. And the first mechanism, both these kinases are recruited to the plasma membrane through their ability to bind to Pi3-45P3 or PIP3, which is the lipid product that's generated by PI3 kinase activation. The second mechanism involves the prior phosphorylation of AKT at serine 473 by the mTOR protein kinase. This creates a docking site for PDK1 to bind to this motif and then phosphorylate threonine 308. And activation of AKT really plays a critical role in driving the growth and proliferation of most cancers. So most pharmaceutical companies will have programs to try and develop drugs that stop the activation of AKT in cancer cells. And the key question is how to do this most efficiently. And what we found actually from our work and many others who work on this pathway is that inhibitors of PI3 kinase, PDK1, mTOR, mTORC1 have all been, or S6 kinase have all been generated. And what's been found is that these inhibitors are only partially effective at suppressing the activation of AKT in cells. And I'm not going to go into all the details here, but what we found recently is the most effective way to stop the activation of AKT in cells is to use a combination of PDK1 and mTOR inhibitors, and because these are the two kinases that mediate the activation of AKT. And it's only when you use these two these combination of inhibitors do you effectively block all the downstream signaling responses that occur in the cell. And this is just one example of a lot of work that's been done in this area, where we look at the critical activating phosphorylation sites on AKT, which are threonine 308 and serine 473, and you only see significant suppression of these phosphorylation sites when you combine the an mTOR inhibitor with a PDK1 inhibitor, and only in the combination of these inhibitors do you get very effective suppression of the phosphorylation of these sites. And and in every cancer cell line this has been done on, you see the same results. So in the course of doing this work, we've also been working with drug companies who work on AKT inhibitors. So there's currently, I checked yesterday, there's actually 284 clinical trials that are either taking place or planned in which AKT inhibitors are being investigated, for the treatment of cancer. And a lot of these programs aren't going, as well as anticipated, because it's what people are discovering is that the AKT inhibitors aren't effectively suppressing this second branch of the, of the signal transduction pathway. But what we've also discovered is that in many cancer cells that are resistant to AKT inhibitors, what happens is that there's a cousin of AKT, and these are enzymes called SGK that become activated. And these enzymes are very similar to AKT, but they're distinct enough not to be inhibited by the AKT therapy inhibitors. And it's the activation of these kinases that have largely been neglected that is driving a lot of the resistance to AKT inhibitors, in the clinic. So we've been doing a little bit more work on these SGK enzymes as a result of this. And what we've also found is even if you take the most sensitive cell lines that are very, very sensitive to AKT inhibitors, and you treat these cells for up to 10 days with an AKT inhibitor, you'll find that very, very quickly the cells start becoming resistant to the AKT inhibitor therapy. And particularly one isoform of SGK called SGK3 starts becoming upregulated. And when this is upregulated, you actually get phosphorylation of its substrate starts appearing in cells. And virtually every single cancer cell line that we've tried, that after you treat them with an AKT inhibitor for up to 10 days, you start getting this upregulation of this SGK3 that counteracts the AKT inhibitor therapy. So the assumption has always been that this isn't necessarily a problem because SGKs are regulated in the same way as AKTs, so by the PI3-kinase pathway and through this pathway. But we started to wonder whether this assumption was actually correct, and we're particularly looking at SGK3, rather than the other two SGK isoforms. And the reason we started questioning this assumption is because, obviously the key messenger that's produced by PI3 kinases is called Pi3-45P3. It's got 3 phosphate groups on the inositol ring, and this actually binds to AKT, has a specific domain called the plexum homology domain that binds to PIP3, and this induces a conformational change that enables it to be activated by PDK1 and mTOR. But SGK3 doesn't have such a pH domain that binds to PIP3, and instead what it has another domain that's called a PX domain. And what this PX domain does, it actually binds to a inositol lipid called PI3P that lacks the 4 and the 5 phosphate group on it. And it turns out that at least in biochemically, SGK3 cannot bind to PIP3, it can only bind to PI3P. And we verified this. Some of these studies were done some time ago, so we've reevaluated all the the old studies that have been done on this and confirmed their data is correct. Wild type SGK3 binds to PIP3. We've also identified 2 critical arginine residues, arginine 50 and arginine 90, that are required for the ability of SGK3 to bind PI3P. And what was quite interesting was that we then looked to see whether binding of PI3P was important for SGK3 activity in cells. And what was really interesting is if you express wild-type SGK3 in cells, it's active and it's phosphorylated at the sites that regulate activity. But if you mutate, the arginine residues that mediate binding to PI3P, you can see you completely lose activity of SGK3. So that tells us that in cells, SGK3 must bind PI3P in order to become phosphorylated and thereby activated. And PI3P in cells is known to reside mainly in the endosomes within cells, and where it plays a critical role in mediating autophagy and protein sorting pathways. And and consistent with SGK3 binding to PI3P, we see that SGK3 is localized at the endosome structures in cells, and when you mutate these arginine residues that are required for PI3 binding, you lose this endosomal localization. So, how do you make PI3P in cells? Well, if you assume that SGK3 is regulated by PI3-kinase, this is the class 1 PI3-kinase, you would need to assume that the PIP3 that's generated from the class 1 PI3-kinase is converted to PI3-4P2 through the SHIP phosphatases, and this is sequentially dephosphorylated by this other inositol phosphate to PI3P. But it turns out that there's other routes in the cell to make PI3P. There's a very obscure class of family of 3 enzymes called class II Pi3-kinases that can phosphorylate PI phosphate to PI3P. Very little is understood about these enzymes. And then there's the much more studied enzyme called class III Pi3-kinase. It's also known as VPS34, which in yeast was first shown to play critical roles in mediating cell sorting pathways. So the key question was, could any of these other PI3-kinases play a role in regulating SGK3? And we were particularly interested in this VPS34 enzyme because it's known to reside on the endosomes where SGK3 is located. And to do this, we've got hold of a compound that's we discovered in a Novartis patent that hasn't been published yet, but we actually synthesized this, this in-house. And this compound turns out to very effectively, dissociate SGK3 from the endosome structures in cells. So this is SGK3 located to the endosomes, and even within half a minute of adding this VPS34 inhibitors to cells, you can see that the SGK3 comes off the endosome. And we've also looked at the effect of this compound on SGK3 activity. And what we find is that the VPS34 inhibitor actually very quickly, within 15 seconds, starts reducing SGK3 activity, and is sort of maximally reduced at about 2 minutes time, and it stays at this level beyond the 2 minutes. So it takes the activity down about 50 or 60%, the inhibiting VPS34. And I'm not going to show you the data. This other bit that comes down, if you combine it with class 1 PI3-kinase inhibitors, it actually comes down to about 80% of the activity. So our feeling is that SGK3s are probably controlled by 2 pathways, by the classical class 1 PI3-kinase pathway, in which the PI3P is mediated through this route, but maybe more importantly by the class 3 PI3-kinases that make PI3P at the endosome. And the reason why I think this is important from a cancer perspective is because this is probably going to be a critical pathway by which cancer cells might escape PI3-kinase therapy by using other pathways to activate SGK3 and other related SGK isoforms that can take over from AKT from driving the proliferation of cancer cells. And and I think the reagents and technologies I've described here will be very useful to further understand what the roles of these class 3 PI3-kinases will be in the cell. And I think I've hopefully acknowledged everybody as I've gone through, showing their pictures or the names after the talks. And I thank you for your attention, and happy to take any questions. Thank you.
Dario, thank you so much for covering so much ground, and also I think revealing that the therapeutic future in this area will mirror the success to date as time goes by. But questions, please. We've got about 5 minutes for questions. Yes, please. Could you wait for the microphone? Thank you.
How widely distributed are the class 2 PI3-kinases in terms of tissue expression?
Yes, I mean, I think I myself have never done any work on the class 2 PI3-kinases, but they are widely expressed, and there's 3 isoforms that obviously differ slightly in their expression patterns, but they are widely expressed and likely to play important roles. Obviously, I think being able now to inhibit the class 3 and the class 1 pharmacological agents does offer an opportunity to disable the other enzymes that make PI3P, and therefore look to see how the class 2 enzymes might be regulated.
Yes, please, Lawrence.
The pharmaceutical industry has gone to enormous amounts of effort, and we still do, in order to get down to highly specific, single enzyme, only inhibits that kinase, not any other kinases. And what that reveals time and time again is that the minute you do that, you actually set off some other parallel pathways, often involving closely related enzymes, that had you perhaps gone for a polypharmacological blur kinase, you might have hit everything at once. Would you comment on whether we should be rethinking that whole drug discovery?
Yes, it's a very interesting perspective actually. It turns out with the AKT inhibitors, most companies when they made their AKT inhibitors actually had agents that did SGK and AKT together, but the manager said no, just make AKT selective compounds. I suppose it's the more things the less selective your kinase inhibitor are, you may start inhibiting undesirable kinases. You know, some kinases that cause general toxicity, and to make your drug less tolerable. I mean, often the advantage of having very specific drugs is you can use them at a very high dose, and to counteract the disease, and that can be very important. That can make the difference between success and failure. The more things your inhibitor inhibits, you'll probably not be able to get such a high dose in patients, and therefore that might restrict the efficacy at which the compounds work. But there are some inhibitors on the market, like Sutent, for example, that inhibit many other kinases, and they're quite effective. But actually, the best example of inhibitors that have been generated recently, that AstraZeneca made a compound called AZD9291 that only inhibits there's the EGF receptors mutated in a lot of lung cancers. And this inhibitor actually only binds to the mutated variety of the EGF receptor, but not the wild type. So it has no side effects. So it only inhibits the cancer cells and not the normal cells. And the hope is that people will start making more of these types of drugs that only target the mutant varieties of the kinases, and that might get rid of the side effects. But it's an interesting question, and it's a very interesting consideration.
Any further questions? Yes, right at the back there.
It was a brilliant lecture. I've been somewhat interested in autophagy because quite a number of the drugs that are toxic interfere with mitophagy, for example. But I've never been able to understand, even looking at your brilliant work on these different pathways, how you produce a stable physiological situation most of the time. You've got a number of feedback systems, yes. But where is the master controller?
So that's an interesting point. I mean, so in the textbook, one always thinks that the upstream, the growth factors and the extracellular signals like oxygen levels, nutrient levels, energy levels, are all coming from the top and regulating the pathway. But in fact, the feedback pathways probably play, an actually more important, or equally as important role. So I don't know if there's just one master controller. I think the pathways sense the system. They have to sense oxygen, nutrients, energy levels. You've got growth factors, and then they have to sense all of these things, and and then interpret what's going on, and then make a decision on whether to switch on the pathway or not switch it on. And I suppose the closest thing you come to have a general regulator of the system is the mTOR protein kinase. I didn't really go into that, but that senses everything, and then it makes a decision, whether or not to be active. So some things switch it on, some things switch it off, and you can have actually a cancer-causing mutation that should be very active, but if you take away, for example, amino acids, or you activate, you reduce energy levels by activating AMP kinase, you can actually switch off the mTOR kinase. So I think the if you're looking for a master control of biological signaling pathways, the closest you get to this is the mTOR kinase, at least for the cancer setting. Probably in the in the Wnt signaling pathways, it's the Wnt kinases themselves that regulate sense ion levels outside the medium through some unknown pathway. And probably in the mitochondrial system that I discussed, it's probably the PINK1 kinase is somehow able to sense the state of mitochondrial polarization through a yet unstudied pathway. But so the sensing mechanisms are really important, and I don't think we understand enough about them. But it's an interesting question.
Any more questions? If not, Dario, it remains to me to thank you for a splendid Sackler lecture, and may we give you a small token, a memento.
Thank you very much.